This chart maps U.S. helminthic infestations. One parasite dominates, showing a massive prevalence peak centered squarely on children 5 to 9 years old. A second correlated spike appears in adults 30 to 39, driven almost entirely by transmission from their infected children. The organism driving these demographics is Enterobius vermicularis, a nematode typically referred to as the pinworm or threadworm. Tracking and diagnosing this widespread infection relies entirely on locating a single microscopic artifact left behind by the parasite. To halt transmission in a clinical setting, physicians have to trace this highly specific biological marker back to its source. Understanding how the disease spreads at a population level requires breaking down the parasite's exact taxonomy, analyzing its highly adapted nocturnal life cycle, and recognizing the unique morphology of its ovum. Helminths is the broad classification for parasitic worms. To isolate Enterobius vermicularis, we divide this group based on structural characteristics. The first division separates flatworms from roundworms, or nematodes. Enterobius vermicularis sits on the nematode branch, specifically maturing within the human intestinal tract. Placing the pinworm in the nematode category tells us exactly what kind of physical structure we are looking for. Nematodes lack segmented bodies, seen in other worm phyla. Instead, they feature a continuous, cylindrical body, tapering at both ends. To survive the highly acidic environment of the human gut, the organism is wrapped in a tough, protective protein layer called the cuticle. This smooth cylinder encased in a durable cuticle perfectly adapts the organism to navigate and persist within the harsh confines of the digestive tract. Within the Enterobius vermicularis species, the male and female worms exhibit stark morphological differences, a biological reality known as sexual dimorphism. The adult female worm measures 8 to 13 millimeters in length. It maintains that slender, thread-like appearance characteristic of nematodes, terminating in a sharply pointed tail. The adult male is drastically smaller, measuring only 2 to 5 millimeters. Its posterior end also differs, featuring a distinct curve at the cloacal opening. Despite the size difference, the internal volume of both sexes is heavily dominated by their reproductive systems, the testis and vas deferens in males, and dual ovaries and uteri in females. The female's internal anatomy is essentially a factory engineered entirely for massive, continuous egg production. This extreme physical divergence and heavy biological investment in reproduction allow the parasite to cycle through a human host population with incredible speed. The life cycle initiates the moment a human ingests microscopic pinworm eggs, often disturbed from contaminated bedding or clothing and inhaled or swallowed. Once ingested, the eggs survive the stomach acid and travel into the small intestine, where they hatch and release active larvae. From the small intestine, these newly hatched larvae migrate downward, settling primarily in the cecum and the large intestine. Within the colon, the larvae undergo a maturation phase, developing into fully grown adult worms over the course of 2 to 6 weeks. It is in this lower gastrointestinal environment that the adult males and females mate. Immediately following reproduction, the male worm dies. The surviving female, now gravid and carrying thousands of fertilized eggs, remains active. The colon serves as a secure biological incubator for this entire maturation and mating process, leaving the surviving gravid female perfectly positioned to execute a singular, highly specialized task. After mating, the gravid female exhibits a specific behavioral adaptation essential to the parasite's survival. She actively migrates outward, traveling down the rectum to the perianal area, a journey she almost exclusively undertakes at night. Upon reaching the exterior perianal skin folds, she deposits thousands of microscopic eggs directly onto the tissue. The physical movement of the worm, combined with a sticky substance coating the eggs, triggers severe local pruritus, or intense itching. Because this occurs while the host is sleeping, the natural, involuntary response is to aggressively scratch the irritated skin. This scratching physically wedges the deposited eggs tightly beneath the fingernails, setting up a direct hand-to-mouth fecal-oral reinfection loop when the host wakes. This entire process, from initial ingestion to the deposition of a new generation of eggs, takes roughly 1 month, with the adult worms living a total of about 2 months. By timing her migration with the host's sleep cycle, The female forces the host's involuntary reflexes to facilitate the rapid transmission, making this parasite prevalent in pediatric populations. Moving from the biological transmission loop back to clinical diagnosis, confirming an active infection relies on capturing this exact moment in the cycle. A definitive diagnosis requires a laboratory technician to identify the specific microscopic eggs deposited on that perianal tissue. Under magnification, the clinician looks for an ovum measuring precisely 50 to 60 micrometers in length. The egg is encased in a thick, colorless wall, providing the structural resilience needed to survive in open air and on household surfaces. But the definitive identifying characteristic is its geometry. One side of the oval is distinctly flattened, giving the egg an asymmetrical D-shape or plano-convex appearance. Locating this specific plano-convex structure on a diagnostic slide provides absolute confirmation that the parasite has successfully completed its loop inside the patient. This flow diagram illustrates how the pinworm's biology is interconnected. The egg's shape ensures survival outside the body, allowing nocturnal migration to force transmission. These massive infection rates exist solely because this adaptation exploits human contact. Clinical laboratory science acts as the bridge here, connecting the physical discomfort of the patient to a rigorous visual confirmation of the organism responsible. By identifying that characteristic plano-convex ovum under the microscope, clinicians expose the hidden chain of transmission, providing the exact data needed to treat the patient and eradicate the most common helminthic infection in the country.